So
Okay.
basically, yes, without any sort of more manipulations, my talk will be a little bit different because I would like to discuss basically a more general sort of view of how eDNA has been used, especially in real-world cases where industries and stakeholders and government agencies have started to utilize eDNA data. So, but let me just start with talking about the problem that we face and what is eDNA going to help us solve at the global scale. Or economies or life and basically or well-being are all linked to nature. And interestingly, like people are learning about this more and more. And more and more, I would say, legislative bodies in different countries are starting to realize that impacting nature is going to cost us. The type of activities that we do in natural resources, in land use, in various types of industries, I think it's impacting the nature that we depend on. And the traditional approaches that we have for understanding changes in nature has not been very effective, especially when you're looking at a more monitoring scenario. We've done a great job, perhaps, documenting some biodiversity, but this monitoring business is different. We need to generate data at a scale. We need to be able to do this repeatedly, and we need to do this cost effectively in a timely manner so that it can become a guide for or other types of activities and developments and so on. This has caused quite a lot of differences and issues, when you're looking at different economies. And I'm giving this example of the yin and yang sort of side of what's happening, for example, in North America or in the US, and what's happening in Europe, which essentially the issue is the same thing, which is it's very difficult to basically track the impact of our activities, on nature in U.S. and North America, especially recently. They have taken this approach of, okay, we cannot measure, so let's just don't bother with environmental regulations and just go in and develop or economy, like we're falling behind. We need these projects, when it comes to oil and gas, when it comes to, like various types of mining activities and so on. In EU, the situation is the opposite. Still, the problem is that how to measure, how to deal with the impact on nature, and they have actually in the past 2 or 3 years, they have legislated a number of different rules, policies like the CSRD, TNFD, various types that will ask the industries to measure the impact of their activities on nature. However, they don't have a robust methodology to do this. Like some people rely on historical data, some people rely on, like modeling, some go with traditional approaches which are not as scalable. So still, like the issue is the same, is a different type of, I would say, plan or problem. The lack of data is basically a huge issue. Now, a few years ago, these are older sort of things, I'm happy to hear barcoding from like one of the previous speakers. This cover of nature is where we introduced sort of the concept of barcoding, and you can see here Linnaeus, the father of modern taxonomy, with sort of modern outfit with a barcode in his hand. Sort of we are giving these new technologies to the hands of scientists, to discover, to identify, to delineate species and biodiversity. But we also need to remember that, like we need tools that can do this in a timely fashion, especially when we're dealing with disasters like the situation that we have, in here in this Time magazine cover, in the Gulf region where there was like a huge oil spill, and we need to understand what's going on, the type of, like impact in nature. So really it's time to kind of now put together, it's really nature time. I kind of use this as an anecdote, say like it's time to really put the nature in the forefront. And we started like in my group and like where I started my work with the Barcode of Life. I was the first postdoctoral fellow in Barcode of Life project in 2003 with Paul Hibert at the University of Guelph, and we built, on the barcoding concept, with adding more technologies. Others, we have seen this happening in Japan with the works that going on with eDNA network and like all the good work that's happening. So we're building on like the kind of the notion of barcoding, now building like capacity to do environmental barcoding, or what we call meta barcoding. And but I tried to go a little bit further than this, and we actually established an entity called eDNAtec, which is specialized in bringing these to the hands of more users and industrial applications. This opened in 2017 in another part of Canada, in St. John's, near the ocean and the East Coast. And then we started building up capacity there. For example, moving from the little sequencing that we used, in my lab, MySeq, to much larger platform like NovaSeq. And then we started establishing a standard operating procedures and bringing in a standardization to this with ISO, for example, and build workflows and so on and so forth. And then the kind of last piece is including some AI type analysis and so on, that I will talk a little bit at the end. So this has been a journey, like the evolution, and we still, like have a long way to go, but that's where, like my side of, like journey has been. I don't want to go into this, the how environmental DNA works. You're all familiar with this, but I like to sort of talk about like all the global projects that's been happening, like under this umbrella of environmental DNA. It's amazing. This is a paper that we published actually like 6 years ago now, but like all of the studies that you can see that basically look at different sites, different groups of organisms, so it's becoming very, very popular in the research side. And but interestingly, most of this work is happening within the academic sector. So this is from the paper. Here is basically the publication year, and it's the number of publications that are published. These are different sectors. You see the academic sector has got the lion's share of the publications, and then comes to government labs, and then industry has a much a smaller sort of portion of this, and that's where I think that needs to change if we really want this to be used in everyday analysis, in impact assessment for industrial activities. I think that we need to move outside academia and look, see how we can transfer this technology elsewhere, in other sectors. This is another sort of global map of all the research that's happening in eDNA. The size of these bubbles represent the number of papers published by a country. You will see Japan is here, Canada is here, United States, and so on and so forth. You see, this is already a very, very much a global effort. And of course, industrial and sort of more, I would say, developed countries contributed more, but we will see that other regions of the world are becoming more and more involved and engaged as well. Now the question is, okay, how do we do this transformation? What will make eDNA an acceptable tool in the real world applications? This is a kind of question that, like I wonder about, and I don't want to go into the details, but I say that a couple of pillars for this are standardization and validations, that needs to happen. I know Christian Meisner is there, having fun in Tokyo, great, and he's going to talk about standardization later. I'm not going to go over it, but a number of validations, needs to happen also, from scientific validation to some demonstrating that is repeatable, understanding the accuracy of the techniques. Like somebody was telling me that, look, I have come up with a new sampling method. Oh, I have a new gadget. You can just dump it in the water and it can sample for you, I would say, okay, how did you measure the sensitivity and the specificity of this method? How did you benchmark it? So these are the type of questions that when you do a paper, and you're curious about something, you may not need all of this. But if you want to get this to the hands of users, and if you want to later on make decisions based on this data, you see, that's really important to have these checks and balances. And as I said, you need to engage your regulators early on as well to understand, the new technologies that's like are like becoming more and more feasible, and they can give you more feedback. And I remember last couple of years ago where I was in Japan, I saw that it's great that the ministries are involved, and helping, with the research community to get these tools going. Now, what we have been doing in eDNAtec, like basically all of the above to some extent. We're trying to stay, focused on the leading edge of these technologies. We have PhD scientists, we have also folks with business degrees and understanding, how to, like make sort of a profitable or at least a sustainable business out of this. We have done so far over 300 projects, and these projects means at least like about 100 samples. We qualify them as a project around that mark. We have a major R&D incubator called SIGA, Center for Environmental Genomics Applications, where we actually do collaborative research with academic sector, with government, or own R&D, and so on. As I mentioned, we were able to achieve ISO certification, and we have been working with others in establishing more standards, more ISO standards for specific parts of the workflow. And we've been involved in publishing and generating quite a lot of data, like and 5 million species detected and so on and so forth. But the heart of this, I think, operation is based on this workflow, the 7-step workflow that we call it EnviroSeek. And this is where I think that we wrap it around an ISO 9001 standard. Basically, it starts from, it's not just about like getting a bunch of samples and running them. If you have a core facility with a sequencer, I mean, that is fine, but doing eDNA analysis for decision making for real world problems actually like has a lot of more layers, from designing the experiments to calibrating, your tools, like how to acquire samples, how to, like what type of sequencing technology to use, and we heard some of these in previous talks. I'm not going to go into the details, but these all need to be, standardized, and you can operate at a scale. Like, it's not that, like sometimes you have to deliver results, in short period of time. So those are all the elements, like we have tried to incorporate in this workflow. And then we have done a number of cases, and applications in different sectors. I'm not going to go over it, but one of the major cases that we always see, like in different applications is impact assessment. You see, from fisheries to oil and gas to mining, this impact assessment is always there. But there are other cases like, monitoring, pathogens, understanding a stock assessment, for fisheries, for example, and so on and so forth. I give you a few examples, not too many, just very short. On the impact assessment, this is a case that we've worked with or a government agency, Environment and Climate Change Canada. Like, I didn't know that you have sites that basically marine sites that you dump the disposal sort of waste in there. They came to us and said there are these sites, in the Pacific region, and basically they wanted to know how this might be impacting, the ecosystem. And as you can see here, we got samples from both reference sites and presumably impacted sites, and you will see that we see different communities. We can easily separate these, and basically, like they have now incorporated eDNA analysis in their workflows for these sites. The other one is a baseline biodiversity. This is done with the mining industry in British Columbia, and again, they wanted to understand if their sites, like may have some species that are endangered or protected species. So we did this sampling from the watershed, basically a larger scale water sampling. Everything that is run off into the water, presumably is going to show up. And we've got quite a lot of, mammal species. They were worried about caribou. That's a protected species that was not found in that region, and then basically address their question. The other one that is becoming a lot more, I would say, like common, is restoration assessment. Throughout the past 200 years of industrial activities, we have basically managed to impact a lot of sites that they now need to be restored. Some of the mining activities, some of the uh, oil and gas sites, and you name it. And these sites needs to be restored to be natural, to look natural at least. We need to understand, what are we restoring here? What are strategies we are using? And eDNA is a fantastic tool to guide restoration management. These are sites in northern Canada, in northern Alberta, that we worked, and again, different restoration strategies versus the pristine and natural, forest that we basically compared with eDNA analysis. The big, I would say, application is on invasive species. I'm not going to go too much into details, but this is sites in our home base in St. John's, around St. John's area, where again we work with the government regulatory agency DFO, and they were sending the samples to us blindly. We didn't know what we were looking for, but we were able to actually find species that they had found to be invasive, and we actually found some that they hadn't found, and they thought, oh, their methods are wrong. We don't have invasive species in these sites. They went again and sampled next year, and they found them with plankton nets and so on. So again, eDNA was an early warning, system here for them to detect these invasive species. One area that I would say that it has a lot of implications, especially with indigenous communities, is the community-based monitoring. We can provide these tools in the hands of indigenous communities, local communities, and basically they can become part of the monitoring activities, especially in remote locations where it's very difficult to send, a team to go there. Like this is a project that we've been running for a few years in northern Arctic region in Nunavut, and we are actually like training high school kids. And the interesting thing is that this is not just for education. We have trained them, we have certified them, so the data is actually like informing the regulatory and policy side. So it's for both for educational purposes, but also for like monitoring and getting real data. Finally, I want to say that like we and others, as you probably know, we're building more advanced tools for data analysis visualization. This is the latest tool that we have developed. It's going to be released very soon. It's called EnviroLinks, and is bringing all of this information in one platform and is adding various analytical capabilities, especially through AI. For example, mapping, visualization, archiving, and so on and so forth, which I'm not going to have time to go over it, but stay tuned. Hopefully next time I'll be going over it, or you can go and visit our site and see it yourselves. Also, a final word, mark your calendars. We have some exciting events. One is a small workshop that is going to happen for the 11th time. So we've been running this for 11 years. So in Italy, in Milan in July, and then the 10th International Barcode of Life Conference, which is a big conference and is coming to Asia, is going to be in Bangkok in November, early November. So please, if you're interested, I think registration for both of these are going to be soon, and I have put the websites for you, and you can always also contact me if you have additional questions. Happy to answer them. And thanks again for your attention. And sorry I couldn't be there in person to meet you guys. Thanks for the opportunity.
Thank you very much, Matt. So we have a few minutes to take questions and comments. So if you have any from floors or online. Yeah, Moritz.
Thank you. Thank you, Mehrdad, for the nice talk. I have a question. Sorry if it's a simple question. I'm new to eDNA. I come from a geochemistry background. So we use a lot of certified reference materials to assess the accuracy of our analysis. So I was wondering if there's something similar for eDNA. If I have new development or new tests and I assess eDNA in different machines, different labs, is there something to compare the efficiency and accuracy of eDNA analysis? Thank you.
Very good question. I think it's definitely, I think that's an area that, like we have been thinking. And on the microbial side, there are some positive controls or some type of mixtures that people use for calibration. For eukaryotes, for the kind of the eDNA on fish or others, we actually are developing a couple of groups within our network are developing, and within our the organization that we started, and Christian is going to talk about. You can go talk to Christian, and he's going to tell you that we have plans for having these basically sort of controls for calibration. But mind you, this is going to be only useful for certain type of calibration. When you're looking at unknown material, it's not that easy to find even ways to calibrate. So it's always comparable and statistical. So one of the areas that we have been working on is also additional statistical, methods like occupancy testing, various types of Bayesian network analysis and so on to be able to make sense of the data. So those positive controls are coming. Some are already kind of being developed, but also I think that it won't take away from doing sound sampling and statistical analysis.